Wow, this place is really cool. Hey Britta, what are you doing here?
Oh, hey, what's up? I was actually just taking a break. I need to get back to the lab. Want to come along?
Yeah, where are we?
So this is the Red Sea Research Center here in Kaus, Saudi Arabia. And so you have all the different marine science labs here, but my lab, the reef ecology lab, is right here.
And what do you work on?
I work on population genetics of coral gobies to analyze their population structure and the genetic diversity.
What are coral gobies?
I have a picture. Do you want to see?
Yes, please.
So this is Gobidon citrinus, and then we have Gobidon fuscoruber and Gobidon irregularis, just to show you some examples. So I'm working with coral gobies of the genus Gobidon, and just like many other cryptic species in the Red Sea, they're heavily understudied. A paper from 2013 described 4 new species of coral gobies, but the follow-up research was very limited. And this is why we aim to fill in this knowledge gap and sample coral gobies from different locations around the Red Sea. We went all the way down south and sampled around the Farazan banks, and we obviously also sampled around Kaust in the center all the way up to Yanbu, and we also aim to go further up north to Duba and potentially the Gulf of Aqaba.
What species do you focus on?
All of them. So the species identification of coral gobies is very difficult due to their life stage coloration and the high diversity of largely similar species, as you can see here. This has already caused confusion among researchers in the past, and this is why our approach is to collect individuals of the whole genus. So since the coral gobies have a limited dispersal range, they usually stick to one host coral. So we expect to find several distinct populations in the Red Sea, and who knows, we also might help resolve species boundaries and find a previously undescribed species here in the Red Sea. But this all depends on the final sequencing results. I actually have a video of me sampling the fish. Do you want to see it?
Of course.
So for the sampling, I am scuba diving and I look out for acropora corals, usually in shallow waters between 2 to 10 meters. I swim around and look in the corals, and when I see my gobies in them, I anesthetize them using clove oil diluted in ethanol. Don't worry, in low concentrations the fish can recover.
Wow, cool. How many fish have you collected so far?
About 140, but I haven't finished my fieldwork yet.
And what do you do with the fish after you collect them?
I take fin clips to allow for DNA extraction.
Can you show me?
Of course. So I have my fish here.
Wow, that's really cool, Britta. And how do you perform the molecular analysis?
So you just saw me taking fin clips, and I'm going to extract the DNA from that later on. And then the next step is to amplify the DNA using PCR. So for the PCR, we will try different primers such as CO1, which is a universal primer for species identification, and also one specifically developed for gobies. Then after the amplification, we need to check our PCR product if the right sequence of the DNA was amplified. And then afterwards, we will send it for Sanger sequencing here in Kaust. When I have the sequences of my gobies, they will be aligned to allow for a calculation of the genetic distance. Finally, we will analyze and visualize the relationships among the DNA sequences of my coral gobies. And that's pretty much it.
Britta, that was great. See you later!